Hi, my name is Shirin, and today I'll be going over various kinds of mutations that could occur in a sequence to alter the results of a translation. To look at the effect of a point mutation, consider this coding DNA. To translate this sequence, I'm going to change all the thymines to uracil first. Then, starting from the star codon, which is AUG, I'm going to separate every 3 nucleotides to denote a codon. Then I'm going to be using the coding dictionary to see what do these codons code for. And this is what we get. Now we will look at some of the mutations that can occur in a DNA sequence. When a single nucleotide is changed or mutated, we call it a point mutation. Here are some of the terms that I'll be using to distinguish between the different effects a point mutation might have on a created amino acid chain. In this example, if you compare the original DNA sequence with the mutated one, you will see that a point mutation has occurred at this spot. A thymine has changed to cytosine. Now, using our coding dictionary, we notice that despite there being a C instead of a T, they both still code for the same amino acid. A point mutation that results in no change in the creation of the amino acid is called a silent mutation. This mutation is also a transition mutation because a thymine, which is a pyrimidine molecule, has changed into another pyrimidine molecule, cytosine. In this example, looking at this mutated sequence, we will see that a mutation has occurred at this spot. The purine molecule A has changed into the pyrimidine molecule, which is a T nucleotide. This is called a transversion mutation. The reflected change in the amino acid chain is an alteration in a single amino acid. Instead of asparagine, now we have tyrosine amino acid. This mutation is also a missense mutation. In this third scenario, a cytosine is mutated to be an adenine. Again, a change in pyrimidine to purine is considered to be a transversion mutation. This transversion mutation has resulted in an early stop codon. A point mutation resulting in a stop codon instead of an amino acid is called nonsense mutation. In this last scenario, a readthrough transition mutation has occurred. This is a point mutation that changes the stop codon that codes for an amino acid instead. And generally, mutations of a purine molecule to another purine molecule, in this case A to G, is considered a transition mutation.